So one thing that we need to talk about is that cells divide. And the reason they divide is they divide to make more. This can be periods of growth. It can also be for repair. Because over time, cells are going to die and they're going to get damaged, and you're going to need to repair them. In these cases, what you want to do is you actually want to make carbon copies of the cell that you had. So you want it to be identical. And that's what we get through mitosis. Okay, and mitosis is actually a very, very small part here of this cell cycle. So the cell cycle is quite large, but then you get this small fraction of time where the cells are actually dividing. So what are they doing in the other phases? The first one of these phases is called G1. And during G1, which is part, you'll notice that G1, S, and G2 are all parts of interphase. During G1, the cell is going to be growing. That's why it's G. Anytime the cell is growing, it's a G stage. And while it's this first growth stage, it's going to be accumulating building blocks. So getting materials that it needs for growth, starting to synthesize proteins and things that it needs, and basically getting ready to grow. If you looked at a cell in this phase, you wouldn't see anything different because this is all really kind of deep under the hood. When you get to the S phase, that S, you can think of it standing for synthesis. So this is when the DNA gets replicated. And you remember when we talked about DNA replication, how we talked about making those corresponding strands. Then we get to the G2 phase, and in this G2 phase, now what happens is you're creating energy stores and duplicating organelles. So at this point, and making proteins. So at this point, what you're doing is you are, the first part, you're kind of getting together what you need to build stuff with. And the second part, what you're doing is you are creating those building blocks and kind of amassing those building blocks so that when we get into mitosis, now we already have what we need in order to divide and create two copies of the cell. So you have a chromosome. And remember, your chromosomes are paired. All right, you get one from dad and one from mom. And when we're duplicating your DNA to make more cells, what we're going to do is we're going to take mom and we're going to make a mom copy. And we're going to take dad and we're going to make a dad copy. Okay? When we make these copies, they actually end up connected together for a short period of time. So what they're going to look at is not these two lines that I've drawn. What they're going to look like is much more of like an X shape. So you're going to have sort of the original, and then you're going to have the copy. All right. What we call these, the original and the copy, is these would be sister chromatids. So sort of like, think of them like identical chunks of DNA. Okay. Those are joined by an area called the centromere. And if we look close at that centromere, so if we were able to kind of zoom into it, what you would see, whoops, is that your centromere actually has one sort of focal point on each of the chromosomes. So here is the centromere, and these focal points are called the kinetochore. And I am really not spelling that right. Let's try that again. There we go, kinetochore. When the cells divide, the cytoskeleton will attach to that kinetochore and use it to kind of rip those sister chromatids apart. Until that point, just to kind of keep things structured so they're easy to move around, these sister chromatids are kind of connected by a very weak molecule called cohesin. And you can think of it as like standing for cohesive. And the reason that these sister chromatids are connected is they're going to be moving around, and it's much easier to move something around that's kind of compact and connected together than it is if you've got a bunch of loose stuff. So this is why the DNA compacts into those chromosomes, and why the sister chromatids are held together by that cohesin.